idea of driving the assembly of soft matter. I'll now talk a little bit about the assembly of DNA and how that is used for genomic applications. So again, we go to this diagram that we had before, and now we have a case with a complex molecule and then a simple solvent. And we look at the flows and the electrical flows and how that can be used to manipulate DNA. All right, so we're going to start by looking at DNA at micro to millimeter scales, and then increase the resolution and eventually look at DNA at angstrom level scale in the context of some intriguing biological applications. So this work is motivated by a method for mapping called genomes. It's called optical mapping. And the way it works is as follows. You take very long DNA molecules, chromosome length, DNA. A single molecule can measure several millimeters. You stretch it on the surface, and then once it is on the surface, you cut it to specific locations using what are called restriction enzymes. So these enzymes can actually recognize the local sequence of DNA, and if they find that sequence, cut it. So you see these little links over here that corresponds to spots that were cut by a specific enzyme. By using different enzymes, different restriction enzymes, you can actually cut at different places, and then from that, you can reconstruct the entire sequence of DNA. Once you have it on the surface, you come in with a robot, and the robot is actually going to read all these molecules as if they were a barcode. That's how simple it is. Now, this actually works really, really well. This is actually better than sequencing. You hear about sequencing all the time, but sequencing is not that useful. Mapping is actually a lot more useful, and we can discuss that perhaps later in the question period. Now, the bottleneck for this entire approach is that of actually elongating the molecules on the surface. Because you have to do it without overlapping any two molecules and without breaking them. Otherwise, you lose all of the information that you were trying to generate. So the problem then becomes one of taking a glob of DNA from a cell, right, and doing something to it so that you actually present the molecules in a manner that's amenable for genomic mapping. Now, to make progress in this area, we're going to have to understand what hydrodynamic interactions are. So for those of you who may not be familiar with that, let me briefly explain what that is about. Assume that you have this negatively charged molecule, DNA, in a solvent. You apply a voltage, a gauge field, and the molecule is going to be displaced from left to right. As the DNA molecule moves from left to right, it perturbs the solvent over here. So that solvent perturbation is actually going to come back and influence the motion of some other section of the DNA. And in doing so, it's going to change the configuration of DNA, and that in turn is going to affect the solvent once again. So you have this highly coupled system, nonlinear, that you need to solve to actually address the dynamics. All right? So we refer to these long-range interactions that propagate through a solvent as hydrodynamic interactions. Now, to parameterize our model, we went back to these experiments by Stoll and co-workers in which they had a solid-state nanoform, and they were trying to sequence DNA by measuring the time that it takes for the DNA molecule to go from the top to the bottom. What they do is they apply voltage at the entrance of a little pore, and this simply pushes the molecule from one side to the other. So here you have a simulation then that tries to reproduce that experiment. So you have a molecule on the left, you have a pore, and you apply voltage just when the molecule is in the pore. So that voltage is going to actually move the side that are here from left to right. So we go ahead then and turn on the voltage. So what you will see, if this was working, was that as soon as you turn the voltage, the molecule goes from the left to the right. Nothing too surprising. However, when you look at the flow field that's generated by the molecule as it moves, you actually find that all of the interesting physics about this process are actually hidden in the solvent. Now, whenever people simulate these systems, they do simple Brownian dynamics simulations, and they ignore all that. They assume they have a simple continuum in the solvent with a single diffusion coefficient. The moment you do a more detailed calculation, you take this content into account, you realize that you were throwing away what's actually responsible for all of the interesting aspects of this system. Okay, so those patterns that you see there are what we call hydrodynamic interactions. All right. So we'll start with a simple model for the system then, in which you have beads connected by worm-like chain springs. The parameters from the spring come from the experiments of Marco and Sigia, where they took DNA and with an atomic force microscope they stretch the DNA and they measure the mechanical constants. So those parameters go into the springs that you see over here. All right? And our contribution then to this problem was to actually come up with methods to calculate those hydrodynamic interactions, to do so in a manner that scales with the molecular weight so that the calculation is tractable, and to do that in the presence of surfaces, because surfaces actually interfere with hydrodynamic interactions. All right? So when you go ahead and take this model then, and start to perform some predictions, this is representative of what you find. On the left, you have a diffusion coefficient for DNA as a function of the length of the molecule in microns. The diamonds are experimental data, and then the circles are the predictions of the model. When you take hydrodynamic interactions into account, you get the open symbols, and you see that the agreement with the experiment is actually very good. If you fail to include these interactions, the scaling with molecular weight is actually incorrect. So that's the first sign that you need to take those couplings into account. Now, these parameters are parameters for bulk solutions. So the next question becomes, can we use these bulk parameters and start to make predictions for DNA when it is confined to small channels? So what you see here then are some of the predictions of the model. This is the diffusion coefficient in a slit channel or in a square channel, normalized by the value of the bulk. And in here, you have the size of the molecule, the radius of generation, normalized by the characteristic spacing of the channel or the square. All right? So there's two interesting things about these results. First of all, you see that even when the confinement is pretty weak, in here, for example, these are channels that are 10 times bigger than the size of the molecule, 10 times bigger. And yet, the diffusion coefficient already feels the presence of the walls. The reason you feel it, even from far away, is because of hydrodynamic interactions. The second interesting thing is that these experiments here were performed after the calculations had come up, and the agreement is actually pretty good. So what this is telling you is that we can use a model that was parameterized in the bulk and use it to guide our designs when we go to small dimensions and confined geometries. All right? So using these models then, we try to develop several designs that would allow us to present DNA in the manner that we wanted. And the one that we eventually settled on is illustrated here. We're going to have large channels going in this direction, and then small nanoscale channels overlaid on the big channels. So these are nanoscale channels that go over there. Here you have an expanded view of the big channel and the nano channel. We're going to apply flow in this direction at a control rate that comes from calculation, and we're going to apply voltage in this direction. And rather than showing you the calculation, just let me show you the experiment. All right, so now you have the actual DNA molecules fluorescently labeled. This is the big channel. Here you have the small channels. And you see how through this process you can actually get this global DNA to go into small channels, stretch the DNA a lot. And then the moment you turn off the voltage, you trap your molecules inside. And you can use these now for genomic mapping. Okay? So this seems to work pretty well. Now, there are a few problems. For example, in here, you see that this is brighter than the rest. Or in here, this section is brighter than the rest. The reason for that, we suspect, is that sometimes, even in these very small channels, we're actually bending DNA. So we need to come up with ways of preventing that. And we think that the way to do that is to load the molecules in a controllable manner. So as sloppy as it is, this right now is the best method for actually mapping human genomes on a high throughput manner. So this is being used right now to look at cancer, for example, where the mutations happen faster than traditional sequence can actually detect them. So you have to rely on these methods. And the goal would actually be to take it from the 30 days that it takes us right now to about 1 hour by leveling the molecules at specific sites and being able to actually get them to go into smaller channels. So how do we do that? Well, the first is this park and load system that we're designing. So rather than just having the channels there, the big channels as we did before, you can actually create these little cuvettes in here where you can park the molecules. And then when the calculations tell you is that upon application of a critical voltage, all of a sudden you're going to get the molecules to go from the cuvettes to the long domains. Okay, so that we did better here. You saw that we went to a critical voltage and they'll go. And somehow, for some reason that we cannot understand even with our models, when you do that, you have a lot less bending of the molecules. The second useful thing that you can do to increase the resolution is to force the system to create these donglets that you see over here. You actually manage to track the molecules by having these big loads out here and out here. You can stretch the molecules even more and prevent, of course, the formation of bands. So that would be even better for mapping. But you need to be able to do it in a controllable manner, and that's proven to be somewhat difficult. And then finally, we think that by controlling the chemistry of the surfaces, you can do even better. But there are things that we don't understand, right? An example is shown here. Right? This material that you see over here was created in exactly the same manner as the material that I'll show you 2 minutes ago, where the DNA was just zipping in and all the molecules were stretched. Sometimes, we don't know why, you get situations like this one, where the molecules start to form these grid-like patterns. They fluctuate back and forth. It's a dynamic process. This is in real time. And we simply do not understand the origin of this process. So things to think about. What is clear from a theoretical point of view is that to address these questions, we're going to need a model that has higher resolution and that allows us to predict what happens at the corners where these models are tightly bound. So if you go to the literature then, and you try to see what models are available, this is what you find. The models that we were using before, many of them developed in our group, work at scales of micrometers. They're all there. Each of the beads has thousands of segments. You find atomistic models at the other end of the spectrum, can force fields. You can actually buy software for it. And they work at a scale of nanometers. But these are very expensive. You actually want to describe phenomena over here for genomic DNA and their useless. And if you look at the scale of interest, the mesoscale, you find that there's actually nothing. Right? So we thought that before we started this work, we had to develop a model that would work at these net scales, and that was able to actually develop, predict the effect of salts, electrostatic interactions, and the mechanical properties at this net scale. So that's what we did. We developed a model to do that, and the key features of the model are illustrated here. So you replace all of these atoms that we have here in atomistic DNA, and you replace each nucleotide with 3 sites: the sugar, the base, and the phosphate. All right, and that's repeated for the system. And of course, different bases have different interactions, so you can actually describe the sequence of DNA. We are going to have the 2 strands of DNA, the minor group and the major group. So all of the geometrical characteristics of DNA are captured correctly by construction. That's put in there by design. All right. Furthermore, these molecules are going to be charged, so we'll have electrostatic interactions, and we'll make every effort possible to make sure that we capture the mechanics at a local level correctly. So this is the molecule that we had before. Now each of these spherical segments, which was on the order of 1 micron, is replaced by all of this DNA that you see over here. And the moment you start looking at the structure, you realize that with these coarse-grained models, we were throwing all of that away. Right? With these better models, you do capture all of these effects. That turned out to be important, as you'll see in a moment, in a biological context. So the mechanical properties of this model that we put together seem to be correct. The experimental persistence there is 40 nanometers. The one that we predict is 40 nanometers. The stretching curves agree with the experimentally determined curves from the Bustamante group. Okay. Now, a very important aspect of this model will be that of predicting hybridization. So the ability of two strands of DNA to recognize each other and come into contact. So we begin by looking at melting curves. When we say melting for DNA, what we refer to is we have two strands of DNA fully hybridized. So double strand of DNA, you heat it up, and at a certain temperature, these two strands come apart, they separate, and that's the melting. So these are melting curves as a function of temperature for different salt concentrations. The melting point will correspond to the middle of this curve. The solid symbols are experimental data from scattering, and then the solid lines are the predictions of the model. And you see that the model predicts both the salt effect and the width of this melting transition. So it seems to be capable of describing melting and hybridization. Okay. Now, to calculate those melting curves, one has to predict the free energy of the DNA as a function of some order parameter. And the order parameter in this context is the number of native contacts between the two strands of DNA. So we take two pieces of DNA, each of them with 15 segments, and we calculate the free energy. The melting temperature will correspond to the temperature at which the beginning and the end of the curve have exactly the same height, the same free energy. And what you find when you calculate these curves is that you actually have these large barriers whenever you form about 2 or 3 contacts. So this is telling you then that the hybridization of the DNA is an activated process. And you need to climb this large barrier before you can actually hybridize 2 strands. All right. So that motivated us then to try to take a deeper look into hybridization. If you go to your biochemistry textbooks, what they will tell you is that the way in which 2 strands of DNA hybridize is through the all or nothing approximation. They say that 2 strands of DNA are very happy to be by themselves, but at some point the 2 ends of DNA meet, and when they meet, the DNA hybridizes, it just closes the zip. And you can go to any textbook of biochemistry right now, and that's what they will say. But when you go to the literature, you actually try to look for calculations or models or hybridization, you find nothing. Right? So we thought, well, let's try to use this model and see if we can confirm that prediction. So what you see here is an actual movie of 2 strands of DNA hybridizing. And again, try to see the 2 ends come together and then it closes. Tell me what you find. It was not the end, was it? Much more complicated than that. And this dance of the 2 molecules, they dance, and at some point something happens very quickly, boom, they come together. But it's not the end. So we thought we wanted to look at that in much more detail to try to understand what's happening. Now, this is of importance in general in material science. People are trying to make all sorts of complex materials by looking at the hybridization of DNA. so there's a reason to try to understand this process. Now, the way you analyze these transition state events is through what is called transition path sampling. So it's just a fancy way of saying we're going to generate hundreds of thousands of trajectories in which two molecules hybridize. And then we're going to analyze all of these trajectories to try to find common themes, right, or key features about the way in which these two molecules come together. Right. We're going to do that, and then we're going to generate contact maps. All right. So these contact maps are going to be probability maps that tell us with which frequency one of the strands comes into contact with the other strand. All right. So this is chain 1, chain 2. These are the specific nucleotides along the sequence. When you see a bar in here, it means that this one, for example, has a high probability of coming into contact with this one at the transition state. All right. We're going to analyze this process for a homogeneous sequence and then for a heterogeneous sequence. And we're going to look at events in the transition state ensemble that have only one contact. Right? We're going to be looking for that first instant in which the two molecules come together at the transition state. Right? And this is what we find. If the sequence is homogeneous, you find that most of those initial contacts occur along the diagonal. All right? A little bit offset from the diagonal, meaning that the two molecules are a little bit out of place, but close to the diagonal. Never at the ends. When you go to heterogeneous sequences, you find that most of the time there's a magical contact, the blue one here, somewhere in the molecule, never at the ends. And the moment this contact occurs, the two molecules hybridize. So very different from what we thought. Right? They come together near the middle, and that depends strongly on the sequence of DNA. As you go to longer molecules, of course, this becomes way more interesting, because now you can have two transition states in different sides of the molecule competing against each other, right? And that introduces all sorts of very intriguing complications, but we don't have time to discuss it right now. All right. You can look at this hybridization on surfaces now. One of the strands is bound to a surface, and the whole process becomes even more complicated because you're restricting space, right? So now it has to come along the diagonal to hybridize, precisely in the middle of the molecule, otherwise this reaction cannot occur. So using all this information, then you can go ahead and try to calculate reaction constants for hybridization. To do so, you're going to divide your calculation into two processes. First, the time that it takes the molecule to diffuse from far away into a nearby interface. And then once you're at that interface, the time that it takes for this molecule to react with the other one in this complicated mass. Right? So when all is said and done, then you can actually calculate reaction constants, right, for different compositions of DNA, different lengths, different sequences. You can compare them to experiment, and you find that the actual constants that we calculate are in pretty good agreement with the experiment. So from that point of view, the moral predictions seem to be reasonable. But again, they're very different from this picture that we had in mind about hybridization. All right. So moving on then, as I showed you a moment ago, we wanted to understand what happens to DNA when you bend it very, very tight. And again, in that case, there's a lack of experimental data. Now, an interesting, unlikely source of data we found comes from biology, where people look at virus packaging. In those cases, the DNA that goes into a virus capsid has to be bent very, very, very tightly, right? And from measurements of virus, we can actually compare to the predictions of the model and back down the mechanical properties of DNA. So what you see here then is a calculation in which DNA is being fed through a motor protein into this virus capsid, right? And consistent with what experiments predict, you see the DNA starting to form these loops around the capsid. It's being packaged more and more and more tightly. On the left and the right, you have the same molecule, except that on the right, this represents the molecule is red when it is hybridized, and it becomes blue or green when it actually melts locally. You find that for the most part these molecules are hybridized, but then at late stages, you're trying to bend it so tightly that it doesn't like that anymore. And it comes apart in the middle. And interestingly, it is more efficient for the molecule to pack in this very tiny spot when it is molten than when it is hybridized. And this came as a big surprise, especially to biologists, because the assumption has been that the molecule is fully hybridized all of the time. Now when you look at the experimental data, you find that the predictions, even when it dehybridizes, are fully consistent with experimental data. In here, for example, we have the spacing between different layers of DNA within the capsid in action as a function of the fraction of the genome that's loaded into the capsid. You see that when it is fully loaded, the spacing is about 2.4 nanometers. Experimentally, they actually measure this distance at 2.5 nanometers. That's consistent. More interestingly, people can actually measure the pressure of DNA inside the capsid. There's some very beautiful experiments by Bustamante and some others by the Norris group at LA. They measure the pressure, and they find that it is between 70 and 90 atmospheres. You take the model that I showed you a moment ago, you calculate the pressure as a function of lower genome, it fluctuates a lot. But when you go to 100%, you find that the pressure is about 92 atmospheres. So again, completely consistent with the experiment. So this tells us, right, that the model is reasonable, and it is telling us that in order to actually achieve those pressures, you need to be dehybridized. Now that's an important concept, and I'm going to come back to it in a moment, in the context of DNA packaging now inside the cell. So in the previous example, you're actually cramming all of this DNA into a tiny little sphere, into a tiny cavity. Right? When DNA is actually packaged in your cell, it's also packed into a tiny cavity, but instead of going inside the cavity, it is wrapped around the cavity. All right? So this is the way it is explained, again, in any biochemistry text. You have your double strand of DNA, your double helix. It wraps around these tiny little proteins, these histone proteins, that measure just a few nanometers, about 5 nanometers. It forms two loops around each histone. Right? And then you have many such histones. You form this collapsed structure, and that's what you see in these TM images. All right. Eventually, all of these arrange themselves in such a way as to give you the chromosome. So these are the various levels of organization that we like to understand. All right. And we have a model that can describe DNA over here. So at the very least, I would like to understand that process. Now, what is really interesting is that, and this is central to biology, right? And biologists have no idea, really, about what's happening here. In fact, these images are taken from one textbook. These images are taken from a different textbook, and these ones from another textbook. These are just artistic renderings of what is happening. And I think it would be just nice to try to understand what's happening in more detail. So that's what we're going to be trying to do. Now, very recently, John Wynn at Northwestern has reported a series of very interesting experiments in which he argues that the way in which the DNA wraps around these histone proteins, these little circles, is governed by the mechanical properties of DNA, by the local sequence of DNA. He says that all of this biology is encoded in mechanical properties. And to biologists, this has been like heresy. They don't like that very much. But he's saying the mechanics dictate the way it's packaged. So he performed some experiments in which he actually measured what is the probability that certain motifs in DNA, which are known to be more flexible or less flexible, are located near these histone proteins. And he finds that these are probability curves as a function of composition. He finds that yes, depending on the chemistry of DNA or the mechanical properties, you have high or lower probabilities. The differences are very small, right? These differences in probability are going from 0.25 to 0.31. So it's not tremendous, but it's clearly there, it's very distinct, it is reproducible. All right. Now, there are some, you can criticize it then on some fronts, and people are doing it, and that's the origin of the debate. We are on the camp that believes that, yes, mechanical properties have an influence on how we can practice this thing, and let me show you the evidence that we have for it. Okay? So what you're going to do then is take this model of DNA that I showed you a moment ago and simply bend it and calculate what is the free energy required to bend the DNA from the stretch domain, right, to this fully bended domain. And we're going to do these calculations for different sequences of DNA. The 4 sequences of DNA are called knock, A-track, flex, and stiff. So they have been designed in order to be very, very stiff, like that one, very, very flexible, like that one. Or we have taken sequences from biology that are known to bend very nicely around histones, or some sequences that are never, ever found around histones. Right? You look at the energy, and these are the curves that you get. So at first sight, you go, well, there's no difference whatsoever. But remember that those probabilities are pretty embarrassing. So, what was I saying? So, those probabilities that we were looking at were small. So on this scale, you don't see big differences, but then here, when you go to this expanded scale, you do see that there's some differences. And the differences in free energy, this is the work that is required to bend this molecule, is consistent with the composition that we had in mind. Right? So this stiff molecule requires high free energy. The flexible molecule requires little free energy. And then the one that you find in the nucleosome is somewhere in between. Right? And this is just the mechanical properties, the local sequence. So that's completely consistent with Williams' hypothesis. Now, more interestingly, right, what you find out is that depending on which sequence you try to wrap around the proteins, the amount of time that it spends hybridized or molten changes dramatically. Right? So what you see in here is in red, right, for different sequences, the probability that the molecule is molten, the probability that it is hybridized to a non-native pair, that is the probability that it's making a mistake, or the probability in green that is hybridized to the right complement. And you find that the one that is near the nucleosomes, the biologically relevant one, has very little blue, most of the time is green. In contrast, the ones that do not bind to nucleosomes, A-track or stiff, actually have a lot of blue in them, right? And quite a bit of red as well. So what this is telling you is that for these molecules to actually wrap around a histone, they actually will have to melt a lot and make many mistakes, right? And the moment you melt the DNA around the protein, it becomes accessible, it becomes a target of enzymatic reactions that create havoc in your system. So you don't want that, you want it hybridized. For it to be hybridized, it has to be the nucleosome sequence, or it has to be a very flexible sequence, right? Because it doesn't spend any time making mistakes. So this provides a confirmation that this hypothesis is actually correct, and that indeed the way in which proteins bind to DNA and the chromatin complex is dictated by the mechanical process. So again, I have used my 45 minutes, so I'll stop here and be glad to answer any questions you may have.